Future combination. So let's start with this one. Nakakalito na, di ba? So if four the following genes are actually linked in chromosome number 5. So if you read the chromosome number 5, those genes were linked. So there are three characteristics that you have to consider. One is pertaining to your A gene. The second would be on your B gene and your C gene. Okay? So the first thing to do with regards to this problem is that you have to identify the highest pointer with regards to the progenies. So look for the number of progenies. Identify the highest pointer with regards to the frequency. So most frequent. What is the most frequent? You have dominant. Okay, you have here dominant A, B, and C, which is 390. And then you have a recessive A, B, and C, okay, which is 374. Did you get that? So the first thing to do is to identify the most frequent progenies. Because this would actually tells you that these progenies are the same as your parental type. Did you get that? Okay. The next thing to do is to identify the least number or the least frequent. Okay? Least frequent, the two most least frequent progenies. So, lagi itong dalawa. Okay? So, you have the list here, which is dominant A, B, and C, which is 5. And then, recessive A, B, and dominant C, which is 8. Okay. The next problem is to identify your single crossover in the region 1, in between the first two genes, and the single crossover in between the last two genes. Take note, you have 3 genes there. Okay? So after identifying this one, you have to know the correct sequence of the gene before you can do that. Know the correct sequence of the gene. How? Okay? These two would actually serve as the DCO. Or, ano nga po ulit yung DCO? Double crossover. So, they are the progenies which contains, or they are actually products of the double crossover. Okay? Let's move on. So, how are you going to know the correct sequence of the gene? Okay? You're going to know that if the product or the sequence in the DCO, if this one, or in the parental, if this one undergoes the double crossover, the products should be the sequence which can be found on the least number of frequency. Did you get that? Okay. So this is the example. If this one, if your parent is actually ABC dominant, and the other parent is ABC recessive, if you're going to have here a crossover, a double crossover, if this is the correct sequence of the gene, the products should be, what are the products? Dominant recessive B and dominant C. The other one will be recessive A, dominant B, recessive C. Okay. Do you have the same DCO with the actual observation? No. So what does it mean? It means that this is not the correct sequence of the gene. Okay? So you can actually replace one gene with the other. Or you can reshuffle the sequence of this particular gene. Okay? So how will I do that? This is a trial and error. Okay, trial and error procedure. So for example, I will place here the A and I have first my C before my D. Okay? So of course, on the other one, I have here recessive A, C, and D. If I'm going to have here a double crossover, therefore, what are the progenies? Dominant A, recessive C, dominant B. Another one, recessive A, dominant C, recessive B. Do I get the same sequence of the genes which can be found in the DCO on the actual observation? Yes. So it means the sequence or the correct sequence of the genes should be ACB instead of A, B, and C. Did you get that one? Because this one is the same as this. You need this one before you proceed to get the progenies of the SCO1 or the single crossover. You need the correct sequence of the genes. Did you get that? But of course, there are some problems that would actually give you the correct sequence already, but there are some problems from which you have to move on the genes, okay, or reshuffle the genes in order to get the correct sequence. Nakakasunod. Okay. Nakakasunod ba? Sigurado? Okay, so let's move